Welcome class. In this session, we'll be discussing regarding the requirements for photosynthesis. So what and all are essential for the plants to perform photosynthesis, that will be discussed in detail in this particular part. And the first requirement for the photosynthesis will be radiant energy from the sun. So we have seen that the sunlight, it is used as a source of energy for the photosynthesis. So here the radiant energy received from the visible sunlight. So it acts as a source of energy for the photosynthesis. And if we consider in case of sunlight, so sunlight is composed of packet of energy called as photons. So according to the quantum theory, so light will be composed of a packet of energy called as photons. And in this photon, the energy will be stored, and that energy present in the photon can be considered as a quantum. And the amount of energy that is present in the photons, so it depends upon the frequency of wavelength of the light. Okay. And generally this energy that is obtained from the sunlight, it is represented by the Greek letter nu, where we also represent the incident solar energy as h nu. So this is how the incident radiant energy will be represented. So that is regarding the source of energy for the photosynthesis. Whereas the next important component required for the photosynthesis is the place where exactly this photosynthesis is occurring. So that is site of photosynthesis. So we all know that in case of green plants, photosynthesis will be occurring in case of chloroplast. So chloroplast is a green colored plastid that is present in case of the plants, which is meant for storage of the chlorophyll pigment or the green pigments. And this chloroplast, it acts as a site of photosynthesis. And if we look in case of structure of the chloroplast, so this chloroplast, it is made up of two membrane. It is a double membrane structures, so which forms the envelope. So that is outer as well as the inner membrane. So this outer and inner membrane, they are continuous, they are smooth, and they are known to be selectively permeable in nature. So these are selectively permeable in nature. And in between these two membranes, so there will be a small minute space that is called as intermembrane space. So that is the space that is present between the outer as well as the inner membrane of chloroplast. And inner to this envelope or inner to the membrane of chloroplast, so we can divide the chloroplast into two distinct regions. One is called as grana and another one is called as stroma. So stroma is nothing but the viscous jelly matrix that is present in case of the chloroplast. It is called as stroma. So it can be compared with that of the cytoplasm of other cells. Okay, so it is a viscous jelly matrix that is present within the chloroplast. And this stroma, it will be containing a double-stranded circular DNA. So it contains double-stranded circular DNA within that, and it also contains 70S type of ribosomes, and it contains enzymes for the dark reaction. So these are the components of the stroma. And here, as enzymes for dark reaction of photosynthesis are present in stroma, stroma is considered as a site of dark reaction. So it is a site for dark reaction of photosynthesis. And here, if we look in case of nature of DNA and ribosome, so it resembles with that of the prokaryotic DNA as well as ribosome, where DNA present is double-stranded and circular, and ribosome is of 70S type. And this ribosome is involved in the synthesis of enzymes that are required for the dark reaction. And here this double-stranded circular DNA, so it controls the synthesis of enzymes and all within the chloroplast. And because of this presence of double-stranded circular DNA, chloroplast is also considered as semi-autonomous organelle. So it is called as semi-autonomous organelle because the partial functioning of the chloroplast, that is synthesis of the enzymes required for photosynthesis, all those will be catalyzed by all those will be regulated by its own DNA, whereas functioning is controlled by the chromosomal DNA. So hence, partially this DNA present in chloroplast control its function. It is also called as semi-autonomous organelle. So that is regarding stroma of the chloroplast. And within the stroma, you can see certain stacked coin-like structures. And these stacked coin-like structures, they are called as grana. And this grana is considered as site of dark reaction. So it is a site of dark reaction. And this grana, what is composed of? So each granum, it will be composed of a coin-like structures that is called as thylakoids. So thylakoids are the structural units of the granum. And within this thylakoid, if you see, so again this thylakoid is a double membrane structure. So it is a double membrane structure with the luminal space within it. So it is composed of two membranes, outer and inner membrane. And inside there will be a space that is called as lumen. So that is the structure of the thylakoid. And in the membrane of thylakoid, we can see the presence of the chlorophyll pigments. So chlorophyll pigments, they are present in the membranes of thylakoid, and hence the thylakoids present in the grana, they will be taking part in the light reaction of the photosynthesis. So that's why grana is considered as site of light reaction. So it is not dark reaction, it is considered as site of light reaction. And if we see in case of grana, so each grana, they are interconnected with each other by means of a structures called as stroma lamellae. So stroma lamellae is the interconnecting structure which connects two adjacent grana together. So that is regarding the structure of the chloroplast, which is involved in the photosynthesis, and it is considered as a main site of photosynthesis in case of green plants. And this chloroplast will be abundantly present in the leaf cells called as mesophyll cells, and in case of other green parts of the plants also. So next is regarding the photosynthetic pigments. So we know that photosynthetic pigments are present in chloroplast. Which are those photosynthetic pigments? What is the role played by these pigments? We'll see one by one. So first important photosynthetic pigment is chlorophyll. So this chlorophyll in case of green plants, it is of only two types, that is chlorophyll A and B. Whereas in case of bacteria, algae and all, we can see the other types of chlorophyll such as chlorophyll C, D as well as E also. So it is mainly seen in case of the algae as well as certain cyanobacteria and all. But in case of higher green plants, only two types of chlorophyll will be there. One is chlorophyll A as well as the chlorophyll B. And among this, chlorophyll A is very important. So chlorophyll A is termed as a universal photosynthetic pigment. So that is because this chlorophyll A, it is present in each and every photosynthetic autotrophs. It may be a cyanobacteria, it may be a algae, it may be a higher plants. So in case of all photosynthetic organisms, so this chlorophyll A, it is involved, it plays a very important role in the photosynthesis, and this chlorophyll A is also termed as primary photosynthetic pigment, or it is also considered as essential pigment of the photosynthesis. So why it is called primary or essential pigment of photosynthesis? That is because, so this chlorophyll A, it functions as a reaction center for the photosynthesis, where it will be taking part in conversion of light energy into chemical energy. So light energy will be converted to chemical energy in the chlorophyll A molecule. So that's why this chlorophyll A, It functions as a reaction center for photosynthesis, and it is considered as the essential pigment or the primary photosynthetic pigment. And if we look in case of molecular formula of the chlorophyll A, so it is written as C55H72O5N4Mg. So this is the molecular formula or the chemical formula for the chlorophyll A molecule. And in case of every chlorophyll molecule, it may be A or B type. So it will be having main parts, main two parts. One is called as pyrrole ring structure, and another one is the phytol chain. So these are the two important components in the structure of chlorophyll molecule. So this pyrrole ring will be having magnesium as a central metal ion, whereas phytol chain, it is a fatty acid chain. So in case of chlorophyll A and B, the difference can be seen in case of this fatty acid chain, where in chlorophyll A, it will be having a methyl group, and in chlorophyll B, it will be having a aldehyde group. So structural details of chlorophyll B we'll see further along with the diagram. And here, coming to the chlorophyll A, it is going to absorb the light in the red region as well as the violet blue region. That is, it can effectively absorb the light wavelength of 650 as well as the 450 nanometer very well, and that's how it will be actively taking part in the photosynthetic light reaction. And next is regarding chlorophyll B. So this chlorophyll B, it is one more type of chlorophyll pigment. So here we can see the structure of chlorophyll A and chlorophyll B. So this particular ring structure, so this is called pyrrole ring. It is called pyrrole ring structure, where you can see the magnesium as a central metal atom, and it will be containing the methyl group as a functional group. And this fatty acid chain, it is called as phytol chain. So this is the phytol chain of fatty acid. So it's similarly in case of chlorophyll B molecule also. So here, in case of chlorophyll B, it is similar to chlorophyll A. But as I said, it will be having aldehyde group instead of a methyl group, and its molecular formula, it slightly differs from that of the chlorophyll A. That is, it has the molecular formula C55H70O6N4Mg. So here one additional oxygen molecule will be present and two hydrogen molecules are lesser compared to chlorophyll A. That is because in chlorophyll A, methyl group CH3 will be there. In case of chlorophyll B, CHO, that is aldehyde group will be there. So that the number of hydrogen will be lesser and oxygen will be more than that of the chlorophyll A molecule. So that is the only difference between chlorophyll B and chlorophyll A. And compared to the color of chlorophyll A, so chlorophyll A, so it is dark green in color. It is dark green pigment. Whereas chlorophyll B, so it will be yellow green in color. So that is the difference between the color of chlorophyll A and chlorophyll B. And similar to chlorophyll A, chlorophyll B also can absorb the blue as well as the red light, okay, which is of wavelength of 650. That is for red light, and blue light is of 450 nanometer. But the efficient absorption will be done by the chlorophyll A molecule. So the second set of photosynthetic pigments that are involved are carotenoids. So what are these carotenoids? So carotenoids are the group of yellow-brown to reddish pigments which are associated with the chlorophylls. And these are also found in case of chloroplast, and they are also seen in case of chromoplast also. So chromoplast refers to, it is a plastid which stores the pigments other than chlorophyll. So these carotenoids are found in association with chlorophylls in chloroplast also, and they are also found in the chromoplast. And these carotenoids along with the chlorophyll B, it is considered as accessory pigments. That is because, so whichever the light cannot be absorbed by the chlorophyll A molecule during conversion of light energy into chemical energy, so the other portion of the light will be absorbed by these molecules, okay, and it will be transported to the reaction center so that efficiently the chlorophyll A molecules can synthesize the, or it can synthesize the chemical energy by converting the light energy. So it boosts up the function of the chlorophyll A molecule. Directly it is not involved in conversion of light energy into chemical energy, but it supplies more light energy to the reaction center which is formed of the chlorophyll A molecule. So that's why it supports the functioning of chlorophyll A. So hence it is considered as a accessory pigment. So chlorophyll B and carotenoids, they are considered as accessory pigments. And in case of carotenoids, there are two types. One is carotene as well as the xanthophylls. So carotenes are generally orange colored hydrocarbons with molecular formula of C40H56, and they can effectively absorb the yellow light region. So they can absorb yellow light, whereas xanthophylls, they are yellowish oxygen containing derivatives of carotenes, okay, with molecular formula of C40H56. Oh, so in addition to carotene, one extra oxygen will be present. And this is yellowish color pigment which can effectively absorb the light in case of the orange as well as the green light region. So they can absorb these two wavelengths of the light. So that is how they supplement the light energy for the chlorophyll A molecule to convert it into chemical energy. So that's why these are considered as a accessory pigments. And with respect to the photosynthetic pigments, so we can derive two types of spectrums. One is action spectrum and another one is absorption spectrum. So what is meant by this action as well as absorption spectrum? So first diagram, this is of the absorption spectrum, where you can see different pigments are represented in the graph, showing that which light can be absorbed by them effectively. So for the A is of the chlorophyll A molecule, okay, which has the absorption spectrum almost near the 450 nanometer as well as the 650 to 670 nanometer. So here that is the absorption spectrum for the chlorophyll A. For chlorophyll B, it will be slightly away. It will be near to almost up to 470 nanometer and all. And here also it will be it will be near to 640 or 650 nanometer. For carotenoids, if you see, so this is for the carotenoids actually. So for the chlorophyll B, if you see, it has an absorption spectrum same to that of the chlorophyll A molecule, whereas carotenoids, they have a absorption spectrum more near the green light region. So that is regarding the absorption spectrum, where we can define absorption spectrum as it is the wavelength of light that is effectively absorbed by the photosynthetic pigments. So that is called as absorption spectrum. So which light is absorbed to the maximum by the photosynthetic pigments, it forms the absorption spectrum. So for chlorophyll A and B, it will be almost near to violet blue light as well as red light will be absorbed effectively. Whereas for carotenoids, it can effectively absorb the blue to green light more effectively. So that is regarding absorption spectrum. And next concept is regarding action spectrum. So this second graph is of the action spectrum. So where we can see the action spectrum for the chlorophyll pigments here. So it will be near the violet blue as well as the red light region. So that is the action spectrum. So action spectrum can be defined as it is the wavelength of light at which the rate of photosynthesis is maximum. So it is the light wavelength at which the rate of photosynthesis is maximum. So that is called as action spectrum. So action spectrum can be plotted by plotting the wavelength of light against the relative effectiveness in photosynthesis. So in order to measure the relative effectiveness of photosynthesis, generally the amount of oxygen evolved, that will be determined in order to determine the rate of photosynthesis. So that is regarding action as well as the absorption spectrum. And in order to determine the action spectrum, so actually various experiments has been conducted. So one was conducted by the E.W. Engelman, and later, in order to explain the role of the wavelength of light in case of photosynthesis, so later the two phenomenon has been explained. One is red drop effect, and another one is the Emerson's enhancement effect. Next is Emerson's enhancement effect. So what is meant by this red drop as well as Emerson's enhancement effect? So Robert Hill, he conducted experiments by using monochromatic beam of light in order to determine at which particular wavelength of light actually the rate of photosynthesis maximum. So when he passed a monochromatic beam of a light starting from the violet to the red, he observed that the rate of photosynthesis, it went on increasing from blue to red light region, and once the wavelength of light beyond the 670 or 680 nanometer has been exposed, or the wavelength of light which is beyond 700 nanometer has been exposed, there was sudden fall in the rate of photosynthesis. Okay, so even though wavelength has been increased, there was fall in the rate of photosynthesis, and that is called as red drop effect. And later, Emerson, he conducted the experiment by the experiment of Robert Hill, and what he did is, instead of using a monochromatic light, so he used the dichromatic light. So which is dichromatic light with the alternative shorter and longer wavelength. And in this phenomenon, when he conducted this experiment, so he used one shorter and one longer wavelength. That is, he introduced the red light, which is ranging from 650 nanometer as well as the 710 nanometer. So these two wavelengths of light has been introduced into the plant, okay, which is a dichromatic light, okay, with two different wavelengths. So when he exposed this shorter and longer wavelength of light together, he observed that instead of fall in the rate of photosynthesis, the rate of photosynthesis it continued to increase. So that is called as Emerson's enhancement effect. And this Emerson's enhancement effect, it generally gives an idea of the photosystem in case of the plants. Okay, so in case of plants also which are undergoing photosynthesis, they will be having two different photosystems. One can absorb the shorter wavelength of light and one can absorb the longer wavelength of light. And when these two are functioning together, then the rate of photosynthesis will be maximum. So that concept has been explained by the Emerson's enhancement effect. And what are those photosystems? What are their components? We'll see one by one. So coming to the photosystem. So this photosystem is also called as a pigment system, which refers to complex of the pigments as well as the proteins arranged on the thylakoid membrane as a functional unit. So photosystem is nothing but it is a system which is comprised of the chlorophyll molecules along with certain proteins. Okay, so which are arranged on the thylakoid membrane. So it forms the photosystem. And in case of each photosystem, there are three different components. One is photochemical reaction center. Another one is antenna system or light harvesting complex. And third one is the electron carriers. So what is meant by this reaction center? So reaction center is the one which is composed of one or more chlorophyll molecules combined with specific protein. So if one chlorophyll A molecule is there, so this chlorophyll A molecule will be surrounded by several hundred proteins. Okay, so this complex together, it forms the reaction center. And it is the main site where exactly light energy will be converted to chemical energy. And next one is the light harvesting complex or the antenna system. So it consists of specifically chlorophyll B, submolecules of chlorophyll A also will be there, and numerous xanthophylls and carotenoids, So which are going to capture and feed the light energy to the reaction center. So that's why chlorophyll A and the carotenoids, they form the accessory pigments. So they capture the additional amount of light energy and they supply it to the reaction center for its functioning. So that is the light harvesting complex. So it harvests the additional amount of light and it supplies it to the reaction center. Whereas the third component is electron carriers. So these electron carriers are associated with the photosystem, which forms the electron transport system in case of photosynthesis. So what is the role of this electron transport system? We can understand when we study the light reaction. So that is regarding the components of photosystem. And based on this, we can classify the photosystem into two types, and all this photosystem will be located on the thylakoid membrane. So the two types, so this is the diagrammatic representation of a photosystem. So this is the reaction center, which is surrounded by the light harvesting complex as well as the electron carriers. Okay. So in case of photosystem, there are two types. One is photosystem 1 and another one is photosystem 2. So how these are going to differ from each other, we'll see. So photosystem 2, it will be present in the inner surface of thylakoids, whereas photosystem 1 present in the outer surface of thylakoids. Okay. And here in case of photosystem 1, in addition to this, so it will be having an enzyme complex called as oxygen evolving complex, which liberates the oxygen during the time of photosynthesis. But in photosystem 1, it is not associated with that oxygen evolving complex. And in case of photosystem 1 and 2, the major difference is in their reaction center. Whereas in photosystem 2, it has a reaction center of P680. So where P refers to photosystem. So 680 means it is the wavelength of light that is absorbed by the chlorophyll A molecule that is present in the photosystem 2. Whereas, so this is in case of photosystem 2. Whereas in photosystem 1, it has a reaction center called as P700. That means the photosystem can absorb the wavelength of 700 nanometer during the time of light reaction. Okay. So that is one important difference. And here photosystem 2, it is involved in the process called as photoreduction, where the reducing agent NADPH will be formed, but not in the photosystem 1. And here this photosystem 2, it takes parts only in the non-cyclic photophosphorylation. So what is the cyclic and non-cyclic photophosphorylation? We'll study in the light reaction. And then in case of photosystem 1, if we see, it takes parts both in cyclic as well as the non-cyclic photophosphorylation. And in case of photosystem 2, it receives electron from the splitting of water molecule, whereas photosystem 1 receives the electron from this photosystem 2. And here splitting of water is not coupled with the photosystem 1 because this enzyme complex is absent in photosystem 1, whereas splitting of water can be seen in case of photosystem 2. So that is regarding the difference between photosystem 1 and photosystem 2. And the main thing you have to remember here is photosystem 2 is comprised of reaction center P680 and photosystem 1 is composed of P700. So that is regarding the components or the requirements for the photosynthesis. Apart from that, the raw materials used will be carbon dioxide, which is obtained from the atmosphere. Similarly, water. So carbon dioxide as well as the water molecule, they act as a raw material for the photosynthesis. So this water will be obtained from the soil water. So that is regarding the components or the requirements for the photosynthesis. So in the next session, we'll be discussing regarding the steps involved in the photosynthesis or the mechanism of photosynthesis in detail.